Hello everyone. In this video, as you can see from the picture I have, is one of the most famous and one of the most brilliant minds in the history of science. In this video we are going to discuss about genetics, an introduction to genetics, about the basic concepts that we should know in order to understand genetics, and probably and hopefully if we further move on, we are going to discuss about law of the independent assortment and law of segregation, and how did it all happen, and how did Gregor John Mendel gave us the very foundation and how foundations of genetics, and how and why he's considered one of the father of genetics. So let's begin. In the first to explain how these characteristics of material, like characteristics, were inherited from parents to their offsprings, and how do you resemble to your little bit to your father, a little bit to your mother, or how does this all happen, goes back to the ancient Greeks. So the ancient Greeks were the oldest people who tried to tell that what causes these kinds of heredity materials to pass on from their father to their offspring. And one of the theories was given by Hippocrates. But called he gave an idea based on his knowledge, based on what he thought, was called pangenesis or pangenesis and nesis. And according to this theory, there are particles, or basically there are particles which are called pangenes. There are particles called pangenes which comes from each part of your body. Let's say like particles detached from each part of your body, such as from your heart, from your lungs, from your intestine, from your hands, from your eyes, everywhere, every part of your body. These particles detach and goes into your genetic material, or your sperm and ova. And these sperm and ova, when they gain those particles, they transfer these characteristics to the next generation, and in the same way the characteristics of your father move further on to the offspring. Now his theory suggested that, well, if you think about it, this is not true, because the genetic material of a human being is called the genetic material of a human being is based on the cells which are called germ cells. And any changes and the particles which were detaching from different parts of the body, or different parts of the body cells or somatic cells, will have no effect on the germ cells. So if you can think of it like, if your father is an athlete, or he's a super athlete, he has a lot of stamina, his muscles are really, like those of our athletes, really strong kind of muscles, and he runs fast, or he does all kinds of sports things which and his body's quite fit. But his son wouldn't when he's born, he wouldn't have these kind of muscular features as his father has. He would be just like a normal kid. Or 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 you can view it as like, if your father is a bodybuilder, or if someone is a bodybuilder, his son wouldn't be a bodybuilder when he's born. He'll be just like a regular normal kid. So the particles which we call pangenes do not have any kind of effect on the germ cells. So according to Hippocrates, that the particles pangenes, detach from each part of the body and goes into your germ cells, and in the same way these traits which from your father are carried down to your offspring. This wasn't making quite sense. So this theory obviously got rejected. So later on, the people started giving a new kind of hypothesis called the blending hypothesis. Blending hypothesis. And what this hypothesis says is that, let's just say you have a yellow flower. No, let's just say you have a white flower and you have a red flower, right? And so when you fertilize them together, so when these two flowers fertilize, you get a pink flower. because if you mix red with white you get a little bit of pink kind of color, so you get pink color. So the characteristics just blend in with each other, like colors, which like how colors blend in. And so but when people started conducting experiments, so if you fertilize a white and a red, you would get a red flower, not and the red would be as red as this one right here. The red would be as red as this one right here. There wouldn't be any kind of the red wouldn't be as light, or the color would be as equal as the previous, as this parent red flower right here. So people started thinking, okay, so what happened to this white flower? Did it just disappear, or did it show its properties in any ways or not? And so the blending hypothesis wasn't telling us a lot of about genetics as well, genetics. But later on, this man right here saved us. Literally, he saved us. He saved genetics, and he's considered one of the fathers of genetics. And what he did, he was actually a monk, an Augustinian monk, who in the 1860s gave us some hypothesis, but from his experiments which he conducted, which laid the foundations of modern genetics. And what he did was he said that that heredity, so how should I put this? Like each character, and character means like for a character means like a heritable feature which you have, and which differs, and which is different from organism to organism. Like for example, in this situation, color, a heritable feature, color. So each character has an heritable feature, or a heritable feature, which we call them as genes. So he said each character's heritable features regain their dominancy, or regain their dominancy over the organisms after fewer generations, and so they do not blend in. The heritable features or genes, he used the word heritable features, which later on came to be known as genes. So he said that the heritable features are, or genes, do not blend in. They are just like marbles. So just like you have different colors of marbles in a bucket, if you mix them all up, they wouldn't mix each other. Each one has its own property. It would retain itself. So the heritable features which are for different characters, or do not blend in or mix up. They end up being dominant in later generations. And how did he came up with this? Because is that he first conducted an experiment on pea plants, and what he did is that he took 7 different kinds of characters, and he studied 7 characters of them. And what were these characters were? Some of them were the shape of the peas, or the color of the peas. Like if you're talking about shape, whether they are round or they are wrinkled, or if you're talking about the flowers, whether they're white, whether they're red, or white or pink, or if you're talking about their let's just say, like, there's branches, what kind are they, axial, or are they growing from side by side? So he conducted 7 different characteristics of these plants. And so, but for the sake of simplicity, we are going to talk about basically on the color part, and hopefully we're also going to discuss about the shapes and their how are they wrinkled, and so we'll get on with that. But what he did was he took a flower. Let's just say he took a white flower. So flower white. So this flower right here, and he also took a red flower. Well, this is not red actually, but you get the idea. And what he did was, first he cut off the male reproductive parts of this red flower. So cut off male reproductive parts. And if you love botany, or you have studied botany, you know that the male reproductive part is called the stamen. So he cut off the immature stamens of the of the red flower, and and he took the pollen grains, or the germ cells, or the sperm, you may view them as. He took the pollen grains of this white flower and injected them in the red flower. And before he did that, before he did this kind of experiment, he first of all made sure that these organisms were true breeders, or these pea plants were true breeds. And what do I mean by that? Well, I mean, by true breed, I basically mean that whatever, well, you can view them as like a true breeder is that, like, when you fertilize this plant by itself, it produces exact copy of its offspring. And by that I simply mean is that if you take this plant right here, this red flower plant, pea plant, and if you self-fertilize them, so the offsprings of this plant would be red too. So it wouldn't be like, oh, after 5 or 6 generations you start having white plants, or white pea plants. So you want to have all the offsprings of this plant should be red. And I used to view it, and personally I used to view it as a true breeder, as if whatever I am seeing should be his genetic constitution. And what do I mean by that? Like, if I'm seeing a plant, if I'm seeing a flower, and if I'm seeing, but my best attempt to draw a flower, well, if you're seeing a flower and it's red, and so you don't want its genetic material to be like, okay, his parents was red and his mother was white, so he end up being red. No, you want that, okay, if I'm seeing it red, I want that both his parent and as well as his mother be red. So you can view that as genetic, as a true breeder that has the same kind of genetic constitution, or in other words, when you fertilize them, you get exactly the same plant. Or if you fertilize them more and more, you get the same color plant, not different kinds of color of plants. So first he made sure that these two plants were true breeders. And what he did was, he first cut off the stamen from this part and put the pollen grains. So did I write pollen? Pollen grains from this plant to, oops, sorry. He took the pollen grains from this plant and sprinkled all over with the brush, sprinkled all over on the female reproductive part of this plant, or which are called the carpels. So female are called the carpels, and most of the people, this is good for review, but the point is not here. We're not talking about the parts of the plant, but it's just for general knowledge and stuff like that. So he fertilized these two plants, the pollen from this, the carpel from this. He fertilized these two plants together, and he ended up having a plant which was red. So he ended up having a red flowered plant. Red, the color isn't the same. So he ended up having a red flower plant. Now, when he fertilized this plant by itself, he got, and when he observed a ratio, and he observed that for every 4 plant, 3 of those plants would be actually red, and one would be actually white. So for every 3 you have red, and for every 3 red plants, red flower pea plants, you have 1 white flower pea plants. And he came up with the conclusion that, oh, so this plant's genetic material does gain dominance after some generation have passed through, and it's to the ratio of 3:1. And he conducted experiments several times, and he always came up with nearly the same amount of ratio right here. So he gave us 4 hypotheses, which are basically the basic knowledge. If you're studying genetics, you have to have this knowledge with you. So everybody basically knows that. And he came up with this knowledge is that first, which we mentioned a little bit earlier, that for every character you have for every character you have a version of a gene, or for every character you have allele which are responsible for inheritance in you. So for every character you have versions of a gene, which are known as, well, I hope you already know it by now, which are known as alleles. So alleles are basically versions of a gene. And what do you might be thinking, okay, what do you mean by a version of a gene? But simply a version of a gene means like in this situation, this one also has a genetic constitution, and this one also had its genetic constitution. It also had the gene or the information for color. This one also had the information of color, but they contain different versions of the same gene. This one contained the gene for red color, and this one is contained for the gene for white color. So they have the same gene, but they have the same different versions of the gene. So different versions of the genes of the same genes are called alleles. So this was his first hypothesis. The second one is that for every character you for every allele you gain it from, for every allele, each allele, no, not for every allele, for each version of a gene you get it from one from your parent and one from your each one you get from one from your mom and one from your dad. So each allele, so each for a specific character, so you have two kinds of genes, right? So for let's just say for eye color, so your mom should maybe blue, but your dad would be black, or not that black, would be brown. So you have two versions of the same kind of gene, but you're getting it from different partners. You're getting one from your father, one from your mother. So for each character of a gene, for each allele, you get it from either from your mom or for your dad, and they may be same or different. So both your mom and dad may be brown, as in this is my case, or my mom and dad, they both had brown eyes, so I have brown eyes. Or some of your parents may be, like, one of them may be blue eyes and one of them may be brown eyes. So you basically gain two kinds of allele, two versions of the gene, but from two parents, one from your mom and one from your dad. And if you're getting the same kind of allele, or in some situation you're getting same color, kind of eye color, then you may be known as homozygous. Homo means same, so this will help you to remind. And if you're getting different kinds of genes, or maybe if you're talking about hair, you're getting from your dad, your mom has silky hair and your dad has a little bit of curly hair, so you're getting two different versions of the gene. So you have two versions of the same gene, so that would be a heterozygous. So the second one basically says that for each version of the gene you get, you get it from one from your basically for each character you have two alleles, one from your mom, one from your dad. If they're both same, they're called homozygous. If they're different, they're called heterozygous. Now, if they are same, and then that's not the problem, because if your mom has curly hair and your dad also has curly hair, then most of the chances are that you would have curly hair. But let's just say if it's different, if your mom has silky hair and the dad has curly hair, then it depends that which kind of character will emerge, which kind of character will come, which will show, and what will be the result of your hair. Will you have curly or brown? So the gene which dominates the other is called the dominant. So if for let's just say if if curly hair dominates the silky hair, then 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 you would have a curly hair. So the curly hair gene is dominant. And since you have two different kinds of genes, so you had curly and silky, but you end up having a curly hair, then you're said to be heterozygous dominant. So these two words are joined together. So you would be heterozygous dominant. Heterozygous. Well, yeah, no. Well, yeah, you'll be heterozygous dominant. And if you have two kinds of allele, which are like the father has blue eyes, your mother has blue eyes, so and you end up having blue eyes. But blue eyes is recessive; it usually don't appears. But since both of you had blue eyes and you end up having blue eyes, you would be homozygous recessive. So recessive means the part of the so and so this would be like homozygous recessive because both of the parents had blue eyes. So let's just say like one of your parents had brown eyes and one of your parents had blue eyes. So and you end up having a blue eye, so this would also be considered as a homozygous dominant. And this one, and if both of them had blue, if your parents also had blue and your mother also had blue, then you would end up having called homozygous recessive. Now the last part is that, well, for each character you have an allele, and each germ cell gets one kind of allele for one kind of character. For a character, he gets one kind of allele. And what do I mean by that? Let's just say your father's gene or somebody's gene has both the characters. Like his gene constitution, if you look at it, it has a brown eyes gene, a brown eyes allele, a version of alkali color gene, and a blue eyes allele. And so he has two kinds of alleles. So his germ cells, each germ cell will have one kind of allele. It's not like that one of his germ cells will have both kind of alleles. Like if you look at one germ cell, you have he has also for brown eyes and for blue eyes. No, it would be like if he has a lot of germ cells in him, and so some of its cells would have only for brown eyes, and some of its cells would have only for blue eyes. So I'm hoping not confusing you right here, but it says that each germ cell will have one kind of allele for the same character. And this law is because alleles segregate or separate during germ cell formation, and germ cell formation is formed from a cell division called meiosis. And this law that you get one kind of allele in your each germ cell for the same character, and this law is actually called law of segregation. Which clearly states that for a character you have two kinds of alleles, right? And if your father's genetic material has both kinds of alleles, but each of his germ cells would contain only one kind of genes, or one kind of version of the same kind of character, one kind of version of the gene for the same kind of character. And these are the hypotheses which Gregor John Mendel gave us, and really useful. And so now let's just focus on our flower example into more detail and try to figure out what he's saying. So now, usually when you see in your books, and so those genes which are dominant usually are shown in capital letters, and those genes which are not dominant or which are recessive are shown with small letters. So here we're going to show. Now I'm going to show you that this form that I just wrote, flowers. Now if you were talking about genetic constitution, how would it be represented? Well, let's just start. And how it happens is that first you have to keep in mind that the flowers should be true breeders. And by true breeders I mean that if you fertilize them by themselves, they would end up having the same kind of offspring. They wouldn't end up with having different kinds of offsprings. And what he does is that, so if this flower is white, so I want its genetic constitution to both be P and P. Well, I selected P because not just what alphabet should I select? P wouldn't be quite well. For red, right? So, all right, let's just select small r. So for capital R would be red and small r would be white. All right. So small r and r. Let's just I'm just making this up. So and r and the and this one should also be a true breeder, which means it's genetic. If it's self-fertilized, it should end up with having the same kind of offspring, which means it should end up with having same red kinds of flowers. So these two are the genetic constitution of cell of two flowers. Now if you notice something, that so these flowers have to make gametes, which their gametes will fuse, and eventually they will have an offspring. So now my question is, it's really simple. So this is a genetic constitution of a cell of a plant, and I'm just talking about just the color here. I'm not talking about the whole constitution part, whole kinds of genes and alleles which it has for each character. We're just focusing on the color part. And and if you're just talking about a cell of and its gene which has its color, so if you're talking about color, it has white and this one has red. Okay, but if you're talking about, this is the somatic cell, all right, so keep in mind that this is the somatic cell. Now if you are talking about the germ cells or the reproductive cells, or so, and if you remember from mitosis, the genetics, the germ cells usually contain half of the information from the original cell, because they go through meiosis and end up with half of the information. So whatever this information is, half of this information would be in its gametes. Yeah, I'm hoping I'm not confusing you. So this, if this is 2, so half of it would be 1. So the gametes, so this is the flower, and this is a somatic cell or general body cell, somatic cell, and this one right here is the gamete, right? And this one right here is the gamete. So the gamete contains the half of the information of the original somatic cell, because it goes through meiosis. And this one right here would be small r, only one r. So this one right here would be r. Now these two are the gametes. Now when you fertilize them, these gametes, when they fertilize, you would end up with having an whoops, sorry, drop my mic. So you would end up with having a r and a small r, right? We're having an r and a small r, right? So now this is the new plant cell. This is the offspring. This is the offspring of these two plants right here. So this plant's genetic material is R and R. And now notice, this is not a true breeder. This one has this genetic material, and this one half of it contains this one, and half of it contains this one. Now when this organism fertilizes, or when this organism fertilizes within itself, then you have then what happens? So now you see that the capital R is dominant, which means that this plant is basically red in color, and this one is red and this one is white. So did the white one disappear? No, it's just recessive. If keep on reproducing, eventually it will end up with some plants having white, and they're offsprings of the red flower, but they end up having a white flower offspring. So what happens is, when it fertilizes itself, so it produces sperms which would contain half, if you're talking about the male reproductive part, or the pollen grains in this situation. So it would have R, since you remember that meiosis occurs and it gets half of the genetic information, so half R would be here. One sperm would contain just R, and the other sperm would contain small r. And you might be thinking, okay, why is he putting in the two different sperms? Because according to the law of segregation, for each character here is color. You have two alleles, and two alleles go to different kinds of germ cells. Because alleles segregate or separate during germ cell formation. Now, so these are the sperm. So since this is the genetic constitution of the flower, which has both sperm, which has both the male reproductive part and as well as the female reproductive part, so it would produce the same kinds of ovum too. And so the ovum would be small r, and one would be one would be capital R. Now these are the sperms, and these are the eggs. So if this sperm and this ovum fuses or fertilize together, you end up having an R and R, which means you end up having a red flower. Okay, cool. This one's right. Now if you fertilize this sperm with this ovum, you would end up with having an R and a small r, which means you would still end up having a red flower. So this is homozygous dominant, which has both dominant characteristics. And I think heterozygous dominant. I'm not sure about this part. I'll actually post it in the description video. If I'll go and check it first, and if I'm mistaken here, I'll actually go and write it in the comment below. And so, but I know there is homozygous dominant and homozygous recessive. I don't know if there's something called the heterozygous dominant. I'll look it up. I actually didn't occur to me. But anyways, if we keep moving on, R and R, these two produces red. R and small r, it produces red too. Now, if you have this small r and this egg or in this sperm, you have R and R. So you still end up having a red flower. Now what happens if you get this egg with this sperm? You end up with R and a small r, which means this plant. So out of 1, 2, 3, 4 out of 4, 3 of them would have a red flower, and one of them would have white flower. So the ratio is 3:1. Right? Now, so this is the ratio which Gregor John Mendel came up, and the reason why he was so precise in the equation was that he was actually a physician, and he was actually a mathematician, and so his mathematics really helped him to come up with these ratios and all this math work. So a really smart person right here, really smart guy. Anyways, so this was his observation and hypothesis, and this was his observation, and this was his experiment. Now so after on he carried out, so he actually did his experiment based on only one kind of difference in the character. He just based it on characters which they differ on only one kind of character, and then this was called a color. So then when he fertilized them, he only did his experiment. So first he did his experiment with the difference of only one kind of character. Now he said, okay, what happens if I do it in a two kind of character? And in a two kind of character, he selected the characteristics of round shaped peas and round. Let's just don't know. How do we? Okay, no, it was right. Round shaped peas, as well as round shaped and yellow in color. And the second one, so now you have two characters right here. Here we had only one kind of character. Now here you have two characters. So, and the second one, he took another plant, another pea plant which had. Now I'm going to show them in smaller alphabets just for keeping the tradition keeping the tradition going on. And the second one was, he had wrinkled peas. So if this was round, this was wrinkled, like this. So the wrinkled and and this, if this one was yellow, this one would be green, so green, right? So you had two of these, and and if you notice, they are also true breeders, which means if you keep fertilizing them by themselves, they end up having the same kind of genetic constitution. Because, and so he took these two plants, and so these two, this is just a general body cell, general somatic cell, and what he did was he took these plants and fertilized them. He did the same kind of experiment, and he ended up, so if you want to know, okay, how many combinations does this general somatic cell will go through if you go through meiosis and produces germ cells? What would it what kind of sperms it would produce? What kinds of pollens it would produce? And so let's just do it. Well, it has 1, 2, 3, and 4, right? So the germ cells will have half of the information in it. So half of it will be 4. So each germ cell will have 2, right? So 2. So there's actually a formula to, if you know the number of combinations of germ cells it can make. It's 2 raised to power n. So all you need to do, and n is the number of half of the genetic constitution it will carry, and it actually works with a combination of, I was reading a part in the combination of germ cells it produce, and it actually works with this one too, and it actually makes it a little bit more easier to figure out what kinds of sperm it will produce. So if you want to know how many kinds of sperms you produce, this one was really easy. This one was, if you just cut it in half, this is the only thing that you get. So here, you have 2. So 1, 2, 3, and 4. So 1, 2, 3, and 4. So if you cut it in half, it would be 2. So 2 raised to times 2 would be 4. So you would have 4 types of sperms, and so you can get 4 different kinds of combination, which means y times this, and y times this, y times this, and y times this, which is it's actually the same thing. So each of your sperm, if you have 4, so you would have 4 times of sperm, or in this case you would only have one kind of sperm with R and Y and R and Y. But this method really helps in, hopefully, when I show you in the next video, when I will be doing some kind of traits. I will be putting some traits and from actually for a human, and then we will see that how this formula really helps us. So eventually you guys end up, we people end up with having one kind of sperm, but this is RY. So the gamete of this one would be RY, and in the same way the gamete of this one would be RY as well. So this is the gametes of these two plants right here. Now what do you do? So according to since our current understanding of this law of segregation and things, we would think, okay, so you have one kinds of alleles, right? So if this is a gene, this is a sperm, and this is an ovum. So so the organism would end up having R, Y, Y, right? So now if R pairs with Y, so now I'm doing this based on our current understanding. So if R pairs with Y, so then the sperms would be, so this new plant sperm would be either RY or RY, right? RY. And since this plant also produces ovum and self-fertilizes, this ovum would also be RY, or capital R small y, or well, no, this R, if this R bonds with this Y, and this R bonds with, and so this one should be the smaller R right here. Let me remove that. Yep. So capital R with small y, or capital R with large Y. Let's just suppose that part. Capital R with large Y. Capital R with large Y, since this was the original pair, and so if, based on our current understanding, if we think, okay, if these two are like buddies, they're like friends with each other, they don't want to get separated apart, even then this genetic constitution, they still end up with having a capital R and a capital Y. Well, let's just say that, and then the second sperm would be small r and small y. Okay. And for this part, so if it produces the same kind of ovum, you would have capital R, capital Y, and you had a small r and small y. Okay, that's makes sense. I wrote these because the original sperm was actually R and Y, so it would produce a sperm R and Y, and it would also produce a sperm. It would also produce a gamete like this one, because one half of it comes from this parent, right? But and you would eventually end up with having RY, 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 small r, small y, RY, RY, and RY, and RY, right? So you still end up with 3:1 ratio. You have 1, 2, and 3. You have 3 plants which are round and yellow. This one is round and yellow. This one is also round and yellow. And you have only one plant which has wrinkled and green, not yellow. Oh, yeah, well, that makes sense. That's perfectly clear. But actually, it's not. Actually, this is not what happens. When Gregor Mendel conducted its experiment, he found out that, oh, each of these alleles try to make the possible combinations, all the possible combinations it could make with the other allele. And what do I mean by that? Well, if you just if you go through this formula which I mentioned, and it quite didn't work with this one. Well, it did work, but all of them were equal. All of them had the same kind of genetic constitution. But if you think of this one, this is 1, 2, 3, and 4, right? So half of 4 would be 2. So 2 raised to power 2 times it would be 4. So there would be basically 4 kinds of germ cells. So since we're talking about sperm and ovum, you might want to add them up, and you would end up with 8. So there would be 4 sperms and 4 ovums. So what kind of possible combinations we could think of? Well, R could form combination with this as well as this, right? And this R could also do the same with these two right here. So the possible combinations would be RY, 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 and RY and R and small y. So now you have 4 kinds of sperms, and since this is the same plant, so you would have same kinds of ovums. Right? So now you have 4 times 4. Now you have 16 different kinds of combination. That's a lot. That's a lot of combinations. Then you could have. So you have RY, and let me draw it in this space. R Y, R Y, and RY, and then in the next part you have RY, RY, and this small RY, and you have R, well, this one's the big RY, and you have small RY. So now if you try to, so this is this sperm and this ovum fuse, so you get the idea. You would end up with RY and RY. If this sperm and this are fused, so you get RY, well, you get 2 R's and Y. So I just keep doing it, and hopefully you get you get what I'm trying to say here is these R and Y. So R, Y, Y, R, Y, R, Y. Now I'm trying to fuse this ovum with this sperm right here. Well, and the second one would be R, R, oops, R, Y, Y, and then the next one would be R, Y, Y, and this one would be R, Y, R, Y. So you're now seeing that we're having 16 different kinds of combinations. So the offspring would be like, out of these would be 16 different kinds of offspring you could have here. And so now this, the amazing part of it actually is that now we are just talking about 2 traits here. Now what if you have like I don't know, like you have so many different kinds of variation. You have basically in human you have 43 different kinds of 46 kinds of 46 chromosomes, and each of them have so many genes on them. Each of them have different, so many versions of the gene. So you could end up with having huge kinds of combinations here. So basically this is it. So keep on just drawing, and this is R, Y, and R, Y. So you could have any. So now the point which I'm trying to make here is that these. So did you guys see the amazing thing here, that each of these alleles, each of these versions of the genes paired when they were producing gametes according to law of segregation, they were, each of these germ cells were having one kind of specific trait for the same kind of version of the for the same version of the for the same character. They were, each germ cell was having one kind of character, but along this way they were being separated. They were being separated independently. I mean, they didn't care, even though their original, this one had a capital R and a capital Y, and this one had a small r and a small y. But look at this. Look at the germ formation produced by its offspring. This capital R is with the small y, but in the original one you didn't have this. The R was with the capital Y, but when his offspring produced the sperm, this R had the small y. Or if you look here, this R is with the capital Y. So alleles, along with having one kind of allele for the same trait in one germ cell, they also separate independently, and this law, we call it law of independent assortment. And let me write that down. Well, this law is called law of independent assortment. And this is this is the law of independent assortment, and hope you found that useful, and see you in the next video. Bye bye.